Hi everyone, I'm Josemary on behalf of GenScript, and today we are interviewing iGEM Ionis. So iGEM Ionis, can you tell us more about your project, Starchlight?
So maybe we can share our presentation.
Are you able to see it?
Yes.
Yes.
Okay, perfect.
Yes.
So our project consists to upgrade brewer's spring grain, which is one of the principal ways of making beer. So we will use the waste of starch that is present in spent brewer grain to degrade it into lactate. And this lactate will be polymerized into polylactate. So our chassis for these two steps is Escherichia coli. So once we have our polymerized lactate, it will be our stock, yes, of energy. And then once we need some energy, we will induce a signal that will degrade this lactate, this polylactate, sorry, into lactate. And this will be extracted extracellularly. And then this lactate will be used by Schwannella in order to produce some electrons that will be used to produce energy. So yeah.
This is the this is a project in a more complex way. I don't know to what extent you want us to complexify the project.
Sure, please.
Okay, as Solène was saying, there is two parts, two bacteria. One bacteria that will be transformed with with some genes that are able to break down the starch into glucose, and this glucose can then be used as a lactate supplier. And this lactate is able to be polymerized thanks to two enzymes. One enzyme that will transform the lactate to lactyl-CoA, and the lactyl-CoA is able to be polymerized. And this polylactate is the energy stock, and will be depolymerized on demand, and will be used by this other bacteria, which is called Schwannella onienensis, as an electron donor. There is techniques that have never been done. That's why our project is that innovative, and also that we have something to show to the iGEM competition is the synthesis of the polymer, which is the polylactate. It has never been done. It has two teams, I think in 2019 and 2013. Has tried to synthesize it, but they failed. And there is the other part, because Schwannella onienensis is a bacteria that can produce electricity, so it is highly interesting for fuel cells study, or even in an iGEM project. So the transformation of Schwannella to improve the electron transfer rate of the reduction of the lactate will be highly interesting. That's for the for the lab work timeline, we have several windows when we want to build our construct first, which will be by the Golden Gate technique, with the primer amplification and putting our sequence in our desired bacteria. Then there is the validation part, where we have to make colony PCR, or even validating the synthesis of the PLA. There is basically three different parts that we have to check, which is the part that degrades starch, the part that is able to polymerize, and the part that is able to depolymerize. Another part will be we will be working on Schwanella to improve the electron transfer, to know what kind of supplements can increase the electron transfer of Schwanella, and what is the best efficient system to improve the electricity in our system.
And I don't know if you want to know more about the human practice, or yeah.
Yes.
Okay. So of course, human practice are also a big part of the iGEM competition, as you may know. So we already plan to do a lot of things. The first one is doing a podcast with a video in collaboration with two of our teachers. Then we already plan experiments for the elementary school. That's the basic things that all of the iGEM teams usually do. And we also have at school some events like afterworks or bake sales. And we also have two partnerships for now with breweries. And we are now collecting their brewers' spent grains, but we also want to have a partnerships to maybe have some beers and sell them to afterworks and things like that, and to make the promotion of the brewery. And we're also trying to do a symposium on synthetic biology that will be on September, maybe. And we will do a synthetic presentation of biology to our first grade in our school that are not in master yet. And for the communication and collaboration, we already met one team from Europe. We also have a mini jamboree with French teams in Toulouse, with all the French teams, and we are, I think, 12 teams, French teams. And we will also do, only for the Parisian team, a meetup that we will probably do next week. And we also try to communicate a lot with other teams if they are working on the same topic as we are. So maybe we will try to collaborate with their parts, their biobricks, if we can reuse them or things like that. And also we already have 3 meetings with our potential advisor. And that's all the communication part.
So in terms of financial side for the moment, so here are our sponsors. So of course you have the official one for iGEM, but we are also doing our own work on the side to get some new ones, such as Promega and Thermo Fisher, who are helping us in order to be able to make our proof of concept as soon and as well as possible. Can you skip the slide, please?
Yeah, sorry.
Thank you. In order of budget, so this is an estimate that we have for now. Since we are still in the beginning stages of our lab, it's an approximation, but seeing last year's budget, etc., we think it's rather true to what it should look like. So I won't detail everything for you, but basically our funds are coming, first of all, from our schools that participate for mostly the iGEM related fees, such as the Giant Jamboree, the registration, etc. We have launched our crowdfunding campaign last week, which is gaining some traction, which is good. We have done some paperwork to get City Hall subsidies. And we're hosting different events, like Chloe said, on our school in order to get some funds to advance in our project. Yeah, so that's it. Maybe you have questions for us, more details you might want on any part of the project.
Well, thank you so much for sharing more about your project and breaking down the methodology and biosynthetic pathways behind Starchlight. So You mentioned that a lot of this system will be on demand. Can you tell us more about the biomechanics that make this system on demand?
The biomechanics that makes the system on demand is based on an optogenetic system. This optogenetic system is able to allow transcription to the transcription in the of the gene that can depolymerize the PLA. So it would be first of all a constitutive polymerization, and after an inductive depolymerization. That will be freed thanks to the blue light in our example.
Thank you for answering that question. My next question is, I noticed that you have a lot of diverse activities for your human practices, and I think you're definitely trying to engage with different parts of the community and different age groups, which is really great. Can you tell us what your overall goal was with engaging different parts of the community, and what sort of impact you're hoping to make?
But I think we wanted to touch the maximum of people because, for instance, in our school, we in the first grade we didn't know a lot about synthetic biology, so it will be great to inform them about it. And also for elementary school, it will be experiments about our project, like to conduct electricity with fruits or potatoes, things like that. So to inform them that a living organism can be able to produce electricity, things like that. And also we wanted to sensibilize a lot of people around synthetic biology, and also about our project to upgrade waste. And
yeah, also would like to sensitize people on the waste, that we can reuse waste easily due to synthetic biology, and that maybe for breweries that we can do things for them, because when we talk to the breweries, they said that we don't know what to do with those spent grain and they just threw it. So yeah.
Yeah, and of course, this competition is an open source competition, which makes it even more interesting scientifically. Okay, this is the human part, but scientifically, this is highly interesting, and even with the human part, collaborating with some teams that can have a similar project or working on the same topics, doing things with them is really awesome.
So going off that, with the goal of educating as much as the community as possible, if your project were to be further developed, what sort of impact do you think it could have on industry as far as sustainability and introducing this new way to generate electricity and energy?
This could be a way to utilize a source of sugar. It can be, of course, brewer's spent grain. We chose that one because it was not that used, and this is for the smallest breweries. It is most often just put in trashes. So we didn't want it. It could be done also with, for example, bakery waste, or even with bread. We can do it with a lot of glucose sources. So that could be a way to produce electricity at a high rate. I'm not talking about 50 milliliters Falcon. I'm talking about something that is wider. It can be a huge batch of 100 liters containing our bacteria and our transformed bacteria and the whole system. So yeah, it has a lot of different view where we can get the glucose source. But for instance, we took some brewer's spent grain because it was not used. So yeah, this is currently the project. Even if we are able to make a prototype, it will be very interesting to develop it further, to have something that is bigger and at least that can challenge in the market.
Yeah, and to jump on that, I think in the very long term, our goal would be to show the industry that it's possible to combat two of the biggest problem at the moment, so waste generation and electricity production, and to be able to enter in a green economy, bioeconomy. Sorry, I don't have a word in English, but to enter in a thematic of entering bioeconomy and green circular economy to regenerate waste and produce our electricity, even if it's at a small scale. It could be a way to show the industry that it's possible.
Do you have anything else that you would like the audience to know about your project?
Maybe synthetic biology interests us a lot. Some of us want to do it as a career. Some other are really interested about being able to contact the other team to participate as with an open source competition. So this is really not a goal, but at least something that that every scientist would be to participate. I would want to participate at least. It is highly enriching and yeah, as a human development and technical development, we could end with at least better skills that we had.
Well, iGEM Ionis, thank you so much for telling us more about your project. And on behalf of GenScript, we wish you the best of luck in the iGEM Jamboree, as well as our sponsorship competition. And thank you so much again for interviewing with me.
Thank you so
Thank
much.
you for your time.
Thank you for your time.